Good evening, student. Today we are going to learn about the biotechnology principle and processes. Okay. First, we have to learn what is called biotechnology. From its name, we understood that bio. Bio means it is related to the living organism, and technology it is related to the physics. Okay, means we combine the living science or life science and physical science, mathematics, technology to produce the some product that is known as the biotechnology. Again, I repeat, we combine the life science and technology to produce the some product. You may can tell, sir, which product we get the antibiotic, we get the different kinds of proteins. Okay, by the help of biotechnology. Okay. Then you look, means you make an thought that biotechnology is the new branch of science, but not that. We making the bread and cart, we prepared the bread and cart so many thousands years ago. So that is also the part of the biotechnology. Why? Because when we prepare the cart, means our mother in my home, in our home, means add the inoculum. Inoculum means little amount of cart we add in the milk, so that in this little amount of curd have the lactobacillus. Okay, this lactobacillus help to convert the milk into the curd. Okay, and bread also, mean for the making of the bread we use the yeast. Okay, yeast just produce the carbon dioxide, and due to this carbon dioxide there have so many pores in the bread. Okay, you understood? Then you look application of formation in product in wine and other alcoholic beverages is also biotechnological techniques. Okay, means we get the alcohol by the help of yeast, Saccharomyces cerevisiae. Okay, so you know the fermentation process. This is one type of anaerobic respiration. Okay, so by the help of anaerobic respiration we get the alcohol. Okay, means means yeast just fermented the sucrose or glucose present in this fruit. Okay, and change into the ethyl alcohol or ethanol, C2H5OH. Understood na? Then you look, two important techniques which enable the development of modern biotechnology: alteration of chemistry of DNA and RNA. देखिए, this is the two main technique. Okay, two important technique, two techniques of biotechnology. One is genetic engineering, second one is the chemical engineering. First I tell what is genetic engineering and what is chemical engineering, then we read. Okay, what is genetic engineering? To alter the DNA and RNA of the host organism. If we go to a picture, suppose this is a bacteria, this is the host organism. This bacteria having the own DNA. This bacteria having own DNA. But if we incorporate the foreign DNA inside the bacteria, it's have own DNA. If we incorporate the foreign DNA inside the bacteria, then what happen? DNA or genome of the bacteria will be changed. When DNA will be changed, from DNA to RNA will form by the process of transcription. DNA to formation of RNA. So DNA and RNA of this bacteria will be changed. Okay, this is known as the genetic engineering. You look, this is known as the genetic engineering. Okay. Then you look, genetic engineering, what language 10? Alteration of chemistry of DNA and RNA to introduced in the host. Host is a bacteria, host may be plant cell, host may be animal cell. Inside the animal cell or inside the plant cell have its own DNA. But when we insert the foreign DNA inside the host organism, then DNA and RNA of this host organism will change. Okay, when DNA and RNA will change, means genotype will change, and genotype too, when genotype will change, phenotype also will be changed. Okay, phenotype means external appearance. When DNA is changed, DNA to formation of RNA is known as the transcription. RNA to formation of protein is known as the translation. बोलो बोलो. Translation means first DNA to RNA is known as the transcription. RNA to formation of protein is known as the ट्रांसलेशन. ओके, तो डीएनए अगर चेंज हुआ, देखिए कोई ऑर्गेनिजम्स का डीएनए चेंज हुआ, इसका डीएनए चेंज हुआ, तो डीएनए चेंज हुआ मतलब डीएनए से आरएनए जो बनेगा वो भी चेंज है, आरएनए बनेगा. आरएनए बनेगा तो फिर आरएनए में चेंजेस आया तो प्रोटीन में चेंज होगा. प्रोटीन में चेंज होगा तो ऑर्गेनिज्म का फिनोटाइप चेंज होगा. अंडरस्टूड. नेक्स्ट यू लुक. That is the next one is the chemical engineering. What is chemical engineering? To maintain the sterile condition. What is sterile condition? Please try to understand. Suppose you change the DNA of those group of bacteria. Okay, you change the DNA of this group of bacteria. You take a petri dish. In this petri dish you give this bacteria. Okay, this bacteria will growing in this petri dish. Okay, so you want only this bacteria is growing in this culture medium. You do not want the from environment, from the atmosphere, any others bacteria add here and grow in this petri dish. You do not want, no. So your product will be contaminated. Anything will be produced in this petri dish. How they will transfer? You take the bacteria. After recombinant DNA technology, you take this bacteria and transfer this bacteria, kept this bacteria in a petri plate. Petri plate what? Petri plate means petri dish. In lab you लोग ना ग्लास मेट ऐसा ग्लास का कटोरा टाइप का होता है, ठीक, उसमें कल्चर मीडियम रखेंगे. यू केप द कल्चर मीडियम मीन्स न्यूट्रिएंट्स मीडियम रखेंगे, ठीक, तो उसमें बैक्टीरिया देंगे. बैक्टीरिया विल ग्रो, बट यू वांट ओनली दोज बैक्टीरिया विल ग्रो इन दिस petri plate, rather than those bacteria, no other bacteria will not grow. So that which product you want from this bacteria, suppose you add the insulin in this bacteria, insulin DNA in this bacteria. So you want only the insulin DNA, insulin product you want, only you want the insulin, so that you want to culture the only desirable microbes or desirable bacteria. Okay, if any bacteria come from environment or atmosphere to this petri plate, your product will be contaminated ना, कुछ और कुछ आ जाएगा. Understood or not? Yes. Then you look, this language means है maintenance of sterile condition, है ना. You have to maintain the sterile condition to enable the growth of desirable microbes. Okay, desirable microbes, which microbes you have given in this petri plate, only desirable microbe you want to grow. Or if you take large quantity, in a large quantity, in a large quantity, definitely desirable microbes will eat the nutrients and they will reproduce. Okay. Or if the host cell is eukaryotic cell, maybe plant cell, maybe animal cell, okay, in large quantity you want to grow only this eukaryotic cell. Okay, for the manufacture of biotechnological product, what I am telling you, want to get only the insulin. You do not want to get any others contamination, means any other mixing of the any product of any bacteria, maybe any poisonous thing has been produced by the another bacteria, so it is harmful for the human society, no, you never want. So that you want to maintain the sterile condition. Sterile means germ free, contamination free. Okay, understood or not? ठीक. Biological product like the vaccine, enzyme, beverages, है ना, drug, etc. Vaccine? Yes, you get the vaccine from. सेकेंड नंबर पॉइंट ये बोल रहा है, आप जो बैक्टीरिया का अंदर में जो आप डिजायरेबल जीन दिए हो, जीन ऑफ इंटरेस्ट दिए हो, आप उसी बैक्टीरिया को ही कल्चर करोगे टू गेट द डिजायरेबल प्रोडक्ट. आपको वैक्सीन चाहिए तो वैक्सीन क्या है, सोचो कोई वैक्सीन चाहिए, आपको कोई एंजाइम चाहिए, एंजाइम प्रोटीन होता है, एंजाइम इज अ प्रोटीन। उस प्रोटीन का जीन किस में दिए हो, होस्ट सेल में दिए हो। होस्ट सेल में देने के बाद तो होस्ट रिप्रोड्यूस करेंगे किस में, पेट्री प्लेट में, तो वो आपका मिल जाएगा। ठीक, दैट इज नोन एज द chemical engineering. Only you want the desirable microbes or desirable eukaryotic cells in this petri plate. Okay, ठीक। नेक्स्ट देखो, gene transfer of the host, gene transfer to host require the vector. What is vector? Vector is the vehicle DNA. Okay, देखिए, suppose what is vector, this is a bacteria, you look, this is a bacteria. Okay, this is a bacteria. You take the plasmid, this is known as the plasmid DNA of the bacteria. Actually, if you look the bacteria have two types of DNA. One is known as the genome, main genome, and rather than main genome, this bacteria have circular double stranded DNA. Double stranded DNA. This bacteria have circular double stranded DNA. ठीक, this circular double stranded DNA of the bacteria is known as the plasmid. Okay, understood what I tell? Bacteria have its own DNA, rather than own DNA, it's have the circular double stranded DNA. ठीक, inside the circular double stranded DNA we can add our gene of interest. What is gene of interest? Suppose आप हमारा पैंक्रियास सेल लियो, पैंक्रियास सेल से पैंक्रियास सेल प्रोड्यूस द इंसुलिन, सो पैंक्रियास सेल का जीन लियो, यू हैव टू इंसर्ट हेयर. ओके, ठीक, देन यू हैव टू जॉइन. सो first this is the circular DNA, first you have to cut this circular DNA by the help of restriction endonuclease. Again I tell, you have to cut this circular DNA, you have to cut this circular DNA by the help of restriction endonuclease. Then you have to join this gene of interest, what you collect from the pancreatic cell. Okay, this DNA is very long, but you only want the small piece of DNA. That is the target gene. Okay, you just टेक द टारगेट जीन. सो पूरा डीएनए को क्या है, इतना बड़ा डीएनए, इतना बड़ा डीएनए, तो उसको पहले काटोगे. आपको जो जितना जीन जरूरी है उतना ही लेना है. यू हैव टू टेक ओनली दिस जीन. देन सो यू हैव टू काट द प्लाज्मिड डीएनए as well as you have to cut the gene of interest. टोटल जीनोम को काटेंगे तो क्या मिलेगा, जीन ऑफ इंटरेस्ट. आपको छोटा सा टुकड़ा मिलेगा, बड़ा डीएनए जो काटे हो छोटा सा टुकड़ा मिलेगा. देन यू हैव टू इनकॉर्पोरेट, छोटा सा टुकड़ों को किस में ज्वाइन करना है, यू हैव टू ज्वाइन दिस जीन ऑफ इंटरेस्ट और जीन ऑफ इंटरेस्ट और टारगेट जीन inside the plasmid. Okay, so that before that we have to cut the plasmid DNA by the help of restriction endonuclease. Okay, then we enter the gene of interest inside the plasmid. तो देने से नहीं होगा, यही पोजीशन में आ गया, लेकिन जोड़ने के लिए कुछ होगा. जोड़ने के लिए क्या है, मॉलिक्यूलर ग्लू है, डीएनए लाइगेज. क्या, डीएनए लाइगेज. लाइगेज मीन्स जोड़ने के लिए यूज होता है. DNA ligase help to join the strand of DNA. This strand and this strand, this strand and this strand. Okay. तो understood. तो क्या हुआ, gene of interest enter inside this. So inside the plasmid. Now this plasmid is known as the hybrid DNA. This plasmid is now known as the hybrid DNA. This hybrid DNA also known as the cDNA. This hybrid DNA also known as the cDNA. Why it is known as the cDNA? Because this part is the part of the bacteria. This part is the part of the human DNA or maybe eukaryotic DNA. Human cell is the eukaryotic cell. So we get the hybrid DNA or cDNA. Then we enter this cDNA in the another bacteria. This bacteria is known as the host. We taken the cDNA and incorporate this cDNA inside the another group of bacteria. Okay, so when this cDNA is entered inside this bacteria, then this cDNA will express, means this DNA to formation of RNA, RNA to formation of protein. तो देखो बैक्टीरिया इस पोर्शन से बैक्टीरिया का प्रोटीन बनेगा, बट फ्रॉम दिस पोर्शन ऑफ जीन इंसुलिन का प्रोटीन बनेगा. आई एम टेकिंग जस्ट एग्जाम्पल, इनिशियल एग्जाम्पल. ओके, सो फ्रॉम दिस पोर्शन protein of the desired protein is produced inside the bacteria body. So we isolate the desired protein from the bacteria and purify it and sell in the market. Understood the total process? When this bacteria will reproduce, means first we insert or incorporate this cDNA in this bacteria. Okay, and when this bacteria will reproduce, formation of so many bacteria. So our एक्चुअली बायोटेक्नोलॉजी का दो टारगेट है. एक ये जो जीन है हमारा जीन ऑफ इंटरेस्ट, इसका कॉपी बनाना. कॉपी बना मीन्स व्हेन दिस cDNA आल्सो ट्रांसफर टू द नेक्स्ट प्रोजेनी ऑफ दिस बैक्टीरिया, इसका नेक्स्ट प्रोजेनी में जाएगा. आवर cDNA विल बी कॉपी along with the cDNA, our gene of interest will copy. Understood? Yes, sir. So this is known as the gene cloning. Cloning means formation of multiple number of copy.
Gene
cloning.
Cloning.
Clone. C-L-O-N-E. Clones. C-L-O-N-E. Cloning. Clone or cloning. Okay. We produce the multiple number of our target gene inside the host cell. Okay, that is the target. And another target, second target is to get the desirable product. Or how we get the desirable product? When this gene or gene of interest will express. What is the expression? DNA to, what is the expression? DNA to formation of RNA is known as the transcription. RNA to formation of protein is known as the translation. तो ट्रांसलेशन होने के बाद जो प्रोटीन बनेगा वो प्रोटीन हमारा टारगेट प्रोटीन है. टारगेट प्रोटीन को प्यूरिफाई करना है एंड वी हैव टू सेल इन द मार्केट. ओके, न्यू लोग है ना. दिस इज द स्टेप्स, बेसिक स्टेप इन्वॉल्विंग इन दिस प्रोसेस. यू लोग आइसोलेटिंग द genomic DNA. Genomic DNA means you take the pancreatic cell from a human body. Okay, then you isolating, how you isolate the genomic DNA? You have to cut. When you cut, then you get the gene of interest or target gene. Then fragmentation of DNA. Okay, so how you fragmentize? By help of restriction enzyme. पूरा जीनोम बड़ा लंबा DNA है, बड़ा लंबा जीनोम है, उसमें जब when you give the restriction endonuclease, then this DNA will cut into small fragment. Then sequencing of DNA fragment को sequencing करना. How you perform the sequencing? By the help of gel electrophoresis. जेल इलेक्ट्रोफोरेसिस वो बोलेंगे है ना, ठीक बाद में बोलेंगे. मीन्स व्हाट हैपन, स्मॉल फ्रैगमेंट विल मूव फास्टर टुवर्ड्स द एनोड एंड लार्ज फ्रैगमेंट विल नॉट मूव सो फास्टर, सो दैट स्मॉल फ्रैगमेंट विल मूव अवे एंड लार्ज फ्रैगमेंट नॉट गो बैकवर्ड. ओके, so this is known as the sequencing of DNA fragment. Then है ना, interaction of DNA है, insertion of DNA in the vector. Okay, then we have to insert, means which fragment is required, which fragment is the gene of interest. We insert this fragment inside the vector है ना. Vector means in this picture what we are looking this in this picture. Okay, this is the vector, sorry, gene of interest. This is the vector DNA. Here this is the vector. We insert the gene of interest inside the vector. Actually vector क्या होता है? Vector is a vehicle. When you come to school, you come by school bus or any car or any bike. Vector bring you from your home to school. Again this vector or vehicle bring you from school to home. Like that, this vehicle or this vector DNA bring the gene of interest, this is the gene of interest, to the host cell. Understood? This vector, why it is known as the vector? Because this thing bring the gene of interest in the host cell. Okay, then insertion of DNA in the vector, then introducing DNA in the host. Okay, we have to introduce the DNA in the host. You look in this picture, this is the host. You look it in this picture, this is the host. Then we introduce this inside the host. Then क्या है culturing the cell. Which cell? Host cell. We have to culture the host cell. You look, this is the host cell. Then when we perform the culture, multiple
numbers of DNA.
host cell produced. Okay. Then transformation of host cell. Transformation of host cell means now DNA or RNA of the host cell become change. Okay, ठीक, and we can make an express DNA and RNA. That is known as the basic steps of biotechnology. Okay, the DNA basic processes of biotechnology understood. Then this is the basic process. Okay, and basic steps also you have understood, hai na? This is the basic steps of the biotechnology. Here 2 points are important. Vectors is the vehicle DNA and plasmid is commonly used vector. It is the small circular double stranded DNA. You look, it is the small circular double stranded DNA self replicating extra chromosomal material of the bacteria. Extra chromosomal means it is, you look, this is the DNA, main DNA, this is the extra chromosomal DNA. Okay, then this is the fast recombinant DNA. This experiment is important. You look, this is the fast recombinant DNA was constructed by the Stanley Cohen and Herbert Bayer in 1972 by linking the gene encoding the antibiotic resistance with plasmid of the Salmonella typhimurium. Okay, first you look, then you give the copy. Okay, then in this copy, otherwise you cannot understood. Okay, then isolation of desirable gene or antibiotic resistance. Cut out the piece of DNA from the plasmid or responsible for antibiotic resistance, which is involved in the molecular scissor or restriction enzyme. Desirable gene or DNA is linked with the plasmid or vector transfer into the host organism. Link of DNA इंट्रोड्यूस द डीएनए लाइगेज. ठीक, देखिए इसमें क्या हुआ, उन्होंने स्टैनली कोहेन एंड हार्बर बायर ने फास्ट बायोटेक्नोलॉजी का एक्सपेरिमेंट किए. ठीक, देखिए लिंकिंग द जीन इन एनकोडिंग फॉर एंटीबायोटिक रेजिस्टेंस विथ प्लास्मिड ऑफ साल्मोनेला टाइफी. साल्मोनेला टाइफी क्या है, कौन सा बीमारी करते हैं, कॉज करते हैं, टाइफाइड. ठीक, साल्मोनेला टाइफीम्यूरम से क्या लिए, व्हिच थिंग आई एम टेलिंग, है ना, मीन्स दिस इज द साल्मोनेला टाइफी. ओके, मीन्स साल्मोनेला टाइफी का क्या लिए, प्लास्मिड डीएनए. वी टेकन द plasmid DNA of Salmonella typhi. Along with this plasmid DNA, we have to insert the antibiotic resistance gene. Antibiotic resistance gene. Then this antibiotic resistance gene have to incorporate in the particular bacteria, that is the yeast bacteria. Okay. So ये जो yeast में अभी antibiotic देंगे ना, antibiotic मतलब which can kill the bacteria. अभी yeast में antibiotic देंगे ना, ये yeast नहीं मरेंगे, because inside this yeast cell antibiotic resistance gene आ गया. एंटीबायोटिक को रेजिस्ट करना मतलब सह सकते हैं, ऐसा जीन आ गया. Again I tell, we taken the cDNA from Salmonella typhi, we incorporate antibiotic resistance gene, then formation of cDNA. cDNA is insert inside the
DNA
and yeast host. Then this host will be antibiotic resistance. This experiment is performed by the Stanley Cohen and Herbert Bayer. This is the first experiment of the biotechnology. Here DNA ligase is known as the molecular glue. Okay, here we have to join that cDNA along with the
molecular glue.
हाँ, हमको तो join करना है ना, we have to join this gene of interest in the plasmid. Okay, ठीक, understood this thing. So this is the basic step. Okay, I hope you understood. Thank you for watching this video.